Okay, so good evening, everyone. Welcome to the second session of Techniques in DNA Protein. Today, we'll start with the introduction. Like I discussed the course curriculum yesterday. The first topic that we had was introduction to some DNA technologies, right? So we'll begin with that and we'll learn why exactly DNA sequencing is needed, and we'll study about the types of sequencing, and we will focus on NGS sequencing. Okay, so that's what we'll be doing today. Um, so yeah, let me just share my screen and show you the, I'll start presenting. Okay.
And calling tools and algorithms for very
I'm sorry, I couldn't hear you. What was it? Hello? Excuse me? Does anyone want to say anything? Okay. Okay, so we'll start with uh, introduction to some advanced DNA technologies. So DNA sequencing technology has evolved a lot over these years. Okay, so first let's look at what exactly DNA sequencing means. So DNA sequencing is the process. Does anyone want to say anything? Okay, kindly everyone keep your mics muted, and if you do have any doubts, kindly keep your hands raised, and after I finish the presentation, I'll just get back to your doubts, so you can just type it out in the chat box, okay? Yeah. So, DNA sequencing is the process of determining the exact order of nucleotides, that is ATCG, in a DNA molecule. This allows scientists to decode genetic information, study variations, and understand how genes function in health and disease. So, as I was giving the introduction to this module yesterday, I spoke about this, right? Like DNA sequencing is nothing but we are going to determine the order of the nucleotides in a DNA molecule. Okay, and it is essential because any variations in these in the order of these nucleotides can cause genetically linked diseases, which can be diagnosed by sequencing. Okay, so it'll allow scientists to decode genetic information and to study variations and understand how genes function in health and disease. Okay, so that is why we perform DNA sequencing. All right, so. DNA sequencing technologies, they have evolved a lot. They've undergone many significant advancements. Okay, so when we were, I hope you all have heard about the Human Genome Project. Have you all like read about that or heard about it?
yes, ma'am.
Yeah. So as you might have read that Sanger sequencing was basically used, right? So HGP was like a huge project which many countries undertook to determine the genetic sequence of humans and many plant and animal species as well. Okay. So that time, it was around 1970s. So they were using a sequencing.
That's the biggest step up. So yeah, I'll show you the little stronger.
Excuse me, who's that? Can you
She got
mute your
more
mic, please?
control with that ball that she sets down.
Okay. Kindly adhere to the rules, please. You're supposed to keep your mics muted unless you have some doubts or anything. Okay? Yeah. So, what? Yeah, as I was talking about Sanger sequencing. So this was the major technology used in the Human Genome Project. So it was a first generation method. It was a very early, a very not very technology based method. Okay, it was very slow, expensive, and it was used in the HGP project. Okay, so this was the sequencing technology used first. Then around 2003 to 5, scientists came up with something called NGS sequencing. Okay, so Sanger sequencing was called as the first generation sequencing. It used dideoxy chain termination to generate DNA fragments of different lengths. It was highly accurate, but it was slow and low throughput. It was used in early genetic studies like for HGP and for sequencing small DNA segments. Okay. Next, around 2003 to 5, there was advancement of a new technique called next generation sequencing. It was a massively parallel sequencing and it enabled sequencing of millions of DNA fragments, okay, simultaneously. It was faster, cheaper, and more scalable than Sanger sequencing. And it was used in genomic research and it is still one of the most commonly used methods till date. It was used in genomic research, personalized medicine, and disease diagnostics. Okay, so this is the method that we'll be focusing on in this course. Okay, next around 2010s, there was third generation sequencing introduced, which is a long read sequencing. Okay, so this a long read sequencing means it could sequence a DNA fragment of which had a large number of base pairs, but NGS and Sanger are like limited to like a number of base pairs. Like they can sequence only a limited number of base pairs of the DNA, but these third generation sequencing methods, they included PacBio SMRT and Oxford Nanopore sequencing. Okay, and as I said, it allows for sequencing very long fragments, unlike NGS and Sanger, making it useful for structural variant detection and epigenomics. Okay, so this is just a roadmap of how different sequencing technologies have evolved over the years, okay? And we'll be focusing on NGS sequencing, okay? Um, so let's look at why DNA sequencing is essential, okay, for diagnostic purposes. So DNA sequencing, as we talked about, it plays a crucial role in diagnostic genetic diseases, okay, and in detecting infectious diseases and even cancer, okay. It helps in identifying mutations, tracking pathogens, and enabling personalized treatment strategies, all right? So the first importance is, as we talked about, genetic disease diagnosis. So it can detect genetic disorders which are caused by mutation, which is any variation in any of the nucleotides. Uh, okay, so suppose if a nucleotide has been substituted by another one, or maybe it's missing from the point where it should be, okay? So that those type of mutations cause variations that will lead to diseases like cystic fibrosis, which is caused due to the mutations in the CFTR gene, and sickle cell anemia, which is caused due to the mutations in HPB gene, okay? So, whole exome and whole genome sequencing. So we learn about this in this course in the further classes, okay? So whole exome sequencing is used for diagnosing rare diseases where symptoms are unclear. So we talked yesterday, right? Like how we know that eukaryotic genes have coding and non-coding regions. So the coding regions are called exons, right? And the whole coding part of a genome is called the exome. So when you have a rare, when you're when you want to diagnose a very rare disease, okay, so you won't waste time by looking at the non-coding sequences, right? So during those instances, whole exome sequencing comes into hand, where only the express parts of the genes are sequenced to detect some rare diseases when the symptoms are unclear, okay? And it helps detect de novo mutations. De novo mutations are nothing but which are new mutations, and these are not inherited genetically, okay? So if a person has a very rare disease which has not been inherited through any generation of his or her family, maybe not from parents or grandparents, and it's completely new, those are called de novo mutations, and sequencing helps to identify these type of mutations as well and treat them, okay? Next, we have cancer diagnostics and precision medicine. So somatic mutation analysis. So it identifies mutations which will drive cancer growth, like mutations in the TP53 gene, mutations in the BRCA1 or BRCA2 genes, which are responsible for breast cancer. Next, we have EGFR genes, mutations. Okay, all these can be detected. It helps in targeted therapy selection. So, as we know, in cancer, they perform chemotherapy to treat cancer, right? So chemotherapy, it is supposed to be very targeted. It should be specific to killing only the cancer cells, right? It is not supposed to harm the other cells. So as we've seen in cancer patient, we can see a lot of side effects after chemotherapy, like hair loss or weight loss. Okay, so to prevent that, we can do something called targeted therapy, in which, unlike chemotherapy, only the cancerous cells are targeted and killed, which will reduce the risk of side effects. Okay, so this is how sequencing can help as well in targeted therapy selection. So if you see, you can sequence the gene, you can identify where exactly the variation has happened, which cell line you're supposed to target. Okay, so that will reduce the risk of side effects. Next, we have liquid biopsy, which is circulating tumor DNA and ctDNA sequencing. So it's going to detect tumor DNA fragments in the blood, and it provides a non-invasive way to monitor cancer progression. Okay. Next, we can detect infectious diseases as well. Uh, Viral and bacterial pathogen detection. For example, the most common example we can take is COVID-19. Okay, so you can detect the genetic material of the virus or the bacteria, okay, like influenza, HIV, and tuberculosis. Okay, that can be done, and you can identify antibiotic resistance genes, and which will help you to choose the right treatment as well. Okay. Next, metagenomics for microbial infections. It is used to sequence the microbiome and detect bacterial or fungal infection. So, if a person is suffering from any severe bacterial or fungal infection, to uh, determine which type of treatment is necessary, you need to know which pathogen has caused the disease, right? So, in those instances, you can sequence the microbiome itself. Okay, so for example, you diagnose gut microbiome imbalances in gastrointestinal diseases. Okay. Next, sequencing helps in prenatal and newborn screening. So we have non-invasive prenatal testing, so which uses fetal DNA in maternal blood to detect conditions like Down syndrome, Edwards syndrome, etc. Okay. Newborn screening, it helps detect metabolic and genetic disorders early for immediate intervention. Okay, so the sooner you detect such diseases by using the fetal DNA in the maternal blood, the sooner you can intervene and help treat those diseases. Okay. Um, so next we have pharmacogenics, which is drug response prediction. It Sequencing can help prepare personalized medicine. Okay, personalized medicine is nothing but medicine prepared specific to the genetic makeup of an individual. Okay, so it's going to take into account all the aspects, all the genetic aspects of an individual, what exactly they are allergic to, or what exactly, the gene needs for it to function. All that is taken into account, and a personalized medicine is created based on that. So DNA sequencing helps determine how individuals metabolize drugs. For example, CYP450 gene variations, they affect a response to blood thinners like warfarin. Okay. So these detections like these will help us determine which type of treatment the person should be given. Okay. So that was the importance of DNA sequencing. Okay. I hope it was clear till here. Any doubts? Anyone? Any doubts so far?
Ma'am, I had a small question, ma'am, about the non-invasive prenatal testing. Uh,
Yeah.
is this the same as the amniocentesis from where they take the DNA from the amniotic fluid, or is it something else?
Yeah, I'm so sorry. I had some internet issues. Can you please repeat your question? It was kind of like my internet got disconnected temporarily.
Uh,
Can you just repeat your question?
yes, ma'am. Ma'am, in the non-invasive prenatal testing, it says that it uses the fetal DNA in maternal blood. Is this like similar to the amniocentesis thinking?
It's not the same as amniocentesis. It's different prenatal tests. Okay, so non-invasive is like a blood test. Okay, and whatever uh, fetal DNA is present in the mother's bloodstream, that can be analyzed. Okay, so for this, all you need is the mother's blood. Okay, so a simple blood draw from the mother is your non-invasive prenatal testing, whereas amniocentesis is where you insert a needle into the uterus, right? That's how it's done to collect a sample of amniotic fluid, okay? So that is how amniocentesis is done. So there are two different things. The way in which they are performed is very different, okay?
Yes, ma'am. Thank you so much.
Yeah, okay. Anyone else? Any other doubts?
Can you tell me what's Edwards syndrome?
Edwards syndrome is uh, I mean, it's a genetic disorder basically. I think it causes it's an extra addition or deletion of a chromosome in the in a fetus, I think. I'm not sure. Okay, let me get back to you, but it's just like an addition or deletion of a chromosome is what it is basically. I'll
Okay.
get back to you on that. Yeah. Anyone, anything else? Okay, so I'm going to go to the next topic if anyone doesn't have any other doubt. So we'll move on to the overview of next generation sequencing technologies. Okay. So just give me a minute. Okay, so next we look at an overview of next generation sequencing technologies. Okay, since this is the most commonly used method and the most high throughput method present currently, we look at this, okay, in detail. So next generation sequencing, it is a high throughput sequencing method that allows for the rapid sequencing of entire genomes or transcriptomes or epigenomes. So it has revolutionized genomic research by enabling massively parallel sequencing. So you can sequence millions of DNA fragments at a time, all thanks to NGS technology, and you can produce millions to billions of reads in just a single run of this sequencing technology. Okay, so this is just a general diagram of the workflow. I'll explain it in detail in the further slides. Okay. Um, so, NGS follows a multi-step workflow. First is obviously the sample preparation. So we'll be extracting the DNA or RNA fragment of interest. We will fragment it into smaller pieces, okay? And then we are going to add some adapters, okay, which will prepare the genetic material for sequencing, okay? Uh, I'll explain this in detail, the procedure in the further slides. So this is just a summary which I'm giving. Next step is will be the library preparation. Okay. We have the DNA fragments and we are going to ligate them with adapters. Okay. So I think you all will get confused. So I'll just go to the detailed workflow itself and then come back to this. Okay. So first we look at the types of NGS technologies. Okay. So there are many types of NGS technologies, but the most commonly used one is Illumina sequencing. Okay, so this is for short read sequencing. Means for like 50 to 300 base pairs. Okay, so for these are called short read sequencing, where you can perform sequencing for like a limited number of base pairs. So in that we have first Illumina sequencing. Okay, so this uses sequencing by synthesis method. We'll look at this as well. Okay, with fluorescently labeled nucleotides, and it produces very short reads, 50 to 300 base pairs with high accuracy and throughput. Um, Applications are in whole genome sequencing, RNA sequencing, exome sequencing. Okay. Next, we have in short read sequencing ion torrent. It measures the pH changes when nucleotides are incorporated. It is faster and cheaper than Illumina, but has higher error rates. So the applications are in targeted sequencing, that is in cancer or infectious diseases diagnostics. Okay. Um, Next type of NGS technologies are for long read sequencing. These will cover a huge number of base pairs, like 10,000 to 100,000 base pairs, and these might be lower in accuracy, but they cover a lot of length of the reads. Okay. Next, so the first technology we have is PacBio SMRT, which is single molecule real-time sequencing. It uses a real-time single molecule approach to generate long reads, and it can directly detect DNA modifications like methylation. Okay, the applications are in de novo genome assembly, structural variant detection, and epigenetics research. Next, we have Oxford nanopore sequencing. It uses biological nanopores to pass DNA through an electric field to detect base changes. It generates ultra long reads, that is up to 1 million base pairs. Applications are in real-time sequencing in remote locations like field work in an outbreak or rapid pathogen identification. Okay, so these are the types of NGS technologies. We will be focusing on Illumina sequencing since it's the most common NGS technology. So we'll be studying this in detail. Okay, and the others are just for your knowledge and reference. All right. So applications of NGS technologies, they are widely used in multiple areas of research and diagnostics. So in genomics, it is used in whole genome sequencing, in exome sequencing, or if you want to detect any structural variants it can be used. In transcriptomics, it is used for RNA sequencing to study gene expression analysis. In cancer genomics, it is used to detect tumor mutations, or liquid biopsy, as we discussed, for cancer detection. Okay. Next, we have in clinical diagnostics, it is used to identify genetic disorders, prenatal screening, and pharmacogenics. Okay. In microbiology and infectious diseases, it is used for microbiome studies, like we study that if any bacterial or fungal infection uh, is there, okay, we can sequence the microbiome and determine which type of microorganism is exactly affecting, okay, in pathogen identification, like in case of SARS-CoV-2. In epigenomics, to study DNA methylation and histone modifications. In evolutionary and population genetics, to study genetic diversity, evolutionary history, and ancestry analysis. Okay, so you can study um, how genes have evolved over a period of time, so you can determine a pattern of any genetic variations all through evolution. And if you want to analyze an ancestry of a person, suppose a person is suffering from a disorder, and you want to uh, you know uh, look at the whole family tree, and determine where exactly this disease was present in the family tree, and where exactly they inherited it from. So it can help in ancestry analysis and genetic diversity in the population as well. Okay, so these are some of the applications of NGS technologies. um, let's look at Illumina sequencing in detail. Okay, so the steps in this sequencing are a little bit complicated. Okay, so I'll be showing you a YouTube video as well now, because if we can't visualize this, it'll be really difficult to understand. So I'll show the video. The video will be going on, and I'll be explaining parallelly as well. Okay, so I'll just split my screen into two. Just give me a few minutes. So since this is difficult to understand, I'm just using a YouTube video as reference, okay? So that it'll be easy for you all to visualize, and I'll be explaining it as well, okay? Um, so Yeah. So Illumina sequencing, it's one of the most widely used next generation sequencing platforms, okay? Because of its high accuracy and scalability, it works on the principle of sequencing by synthesis. So this is the principle used for this sequencing method, where each nucleotide is incorporated into a DNA strand, and those nucleotides are detected in real time using fluorescent dyes, okay? So as you all know, if any a biomolecule is, it contains fluorescent dyes, it emits a specific wavelength of light which can be detected, right? So that principle is used here. So the steps in Illumina sequencing are, the first step is your library preparation, okay? So you will choose the DNA. Suppose, let's take a case here where, suppose you want to detect a DNA, a genetic variation in a person suffering from a genetic disease, okay? He's showing symptoms of some genetic disease, and you want to determine exactly which exact disease, and which uh, part of the gene has the variation been found. Okay, so first you will uh, obviously obtain the DNA or RNA from the individual. Okay, Okay, so yeah, sorry. Library preparation, yeah, so you will extract the DNA or RNA, okay, and there are different methods to do this. First is obviously by blood, by whole blood, plasma, or serum, okay. For that, from that you can extract a DNA or RNA from those biological samples, or or you can use saliva or buccal swabs, okay, uh, which is used for non-invasive genetic testing, or you can take the tissue biopsy. You can use tumor or skin or muscle, okay, or in case of prenatal samples, you can use amniotic fluid, okay. Spinal fluids also are a biological sample to, extract DNA or RNA, or urine and fecal samples as well, okay. So after extracting those samples, there are methods for, purifying DNA and RNA. So it's all purified using maybe like phenylchloroform extraction or a column-based RNA extraction or whatever it is. That is done. Okay. Um, and that's how the RNA or DNA sequence will be collected. Okay, and it will be sorry, it'll be fragmented into small pieces, that is about 200 to 600 base pairs, because as we saw, Illumina sequencing is like a low sequencing technology, right? So it will cover only like a limited number of base pairs. So we fragment the whole DNA into smaller pieces, and adapters are added to both the ends of the fragments. Okay. So adapters are nothing but they are like short DNA sequences which are attached to the end of DNA fragment before sequencing. Okay, and these are essential. Okay, uh, when we talk about adapters, it is very essential in sequencing technologies because it it'll help to bind the DNA to the sequencing platform. Okay, so as you'll get to see in this video, um, you will understand that this whole sequencing is being performed on a machine, okay, on a sequencing platform. So adapters will help to help the DNA fragments to bind to this platform, okay. So it'll also allow amplifications, um, binding to the flow cell, amplification of fragments, and it will also provide sequencing priming sites. Okay, you'll understand why these three are necessary as we move forward. Okay, so the first step is you fragmented the genetic material and you're adding adapters to the ends. Okay, and it should contain sequences that will allow these three criteria to be matched. Okay. So let's look at the video so that you will get like a reference. So first is your sample preparation. Okay. So the adapters are being added to the ends as we talked about. The adapters are being added to the ends and a whole library is prepared. Okay. Next step is cluster generation, which is also called bridge amplification. Okay. So the prepared library of fragments that we have, which we have ligated with adapters on both ends, it is loaded onto a flow cell. So this is how a flow cell looks like. It is a glass slide with laths coated with oligonucleotides, which are complementary to the adapter sequences. Okay. So I obviously you all know what complementarity means, right? So these lanes here in this flow cell are already coated with some oligonucleotides. Oligonucleotides are nothing but short DNA fragments which are complementary to the adapter sequences present in the fragmented DNA. Okay, so these are already coated with that. So the prepared library is loaded onto these flow cells. Okay, so each DNA fragment binds to the oligonucleotides on the flow cell. Okay, since they are complementary, uh, they are going to bind to each other. Okay. So as you can see in this video, the oligonucleotides are already attached to the surface of the flow cell. And now we are adding our prepared library. Okay. Prepared library, when it is added, it's going and binding to its complementary oligonucleotide on the flow cell. Okay. And each DNA fragment, it has binded to the oligonucleotide on the flow cell. The fragments are amplified using a technique called bridge amplification. Okay. So this DNA fragment which has binded to this oligonucleotide, it will form a bridge. Okay. As you it'll come ahead in the video. It's going to form a bridge by hybridizing to the nearest oligonucleotide. Okay. Yeah, I'm sorry. So the DNA fragments are going to be amplified. First, these DNA fragments are going to bend over and they will attach. So the DNA fragment which we prepared, both the ends were ligated with different adapters. Okay, different adapters each of the adapters are complementary to different oligonucleotides. So one end was binded as soon as we put the flow cell, as soon as we put the DNA fragments inside the flow cell. The other end ligates, it bends over like and forms a bridge and ligates to the nearest oligonucleotide. Okay. Next, DNA polymerase, it copies the template and it has created a double-stranded DNA molecule. Okay. So DNA polymerase was added as well. And DNA polymerase, as you know, helps in DNA replication. So it has generated a double-stranded DNA molecule, and denaturation will separate those strands, leaving single-stranded DNAs which are bound to the flow cell. Okay, so we have two single-stranded DNAs which are bound to the flow cell, and this process repeats a lot of time, forming clusters of identical DNA fragments. Okay, so this process, it is being performed again and again, as you can see here. So there are a lot of clusters of identical DNA fragments formed here. Okay, so you can ask why clusters, because they amplify the signal, ensuring that each fluorescent signal is strong enough to detect. All right. Next step is sequencing by synthesis. This is the main principle of this whole Illumina sequencing. Okay. So in sequencing by synthesis, a sequencing primer, this small primer, okay, it's going to bind to the adapter region of each fragment. Okay, so four fluorescently labeled nucleotides, that is uh, ATGC, are with reversible terminators are added along with DNA polymerase. Okay, so to this flow cell, we are adding four fluorescently labeled nucleotides with reversible terminators. So they contain reversible terminators. So whenever DNA polymerase encounters a fluorescent nucleotide, it'll just stop the replication. It is going to terminate the replication. So that is why reversible terminators are added along with DNA polymerase. So DNA polymerase is going to replicate this DNA. It incorporates one nucleotide at a time, as you can see here, right? So it's incorporating one by one. So now, as soon as the fluorescent nucleotide has been detected, it stops the replication. Okay. It's going to terminate there because we've added reversible terminators. Okay. The terminator group ensures that only one nucleotide is added at a time. Okay. Now that this G, which as you saw, it had a fluorescent dye, right? So now it has detected or emitted a specific color, okay, when excited by a laser. So by imaging, we detect this um, wavelength of light, okay, and the terminator and fluorescent dye are cleaved, allowing the next cycle to begin. So this same cycle is going to be repeated for a defined number of cycles, okay, and the sequence is read base by base. So here, whenever so each of these have characteristic colors. Okay, suppose A has a blue fluorescent dye, T has a green fluorescent dye. So every time one by one these fragments are added, it is detecting, I mean, it is emitting a specific color of light, right? So when blue light is emitted, you know that there is adenine present at this position of the DNA fragment. Suppose when green is emitted, you know that guanine is present. Okay, so that is how we are determining the sequence of the strand. Okay, so if A is binding, we know that T is present at the other end. So that is how we are sequencing this whole DNA. Okay. So if you zoom into the flow cell, this is how we are going to visualize. Okay. So there's obviously like a whole system to detect this. Okay. So this is how the wavelengths of light are going to be emitted. So each color will represent um, a specific nucleotide, okay? And all this is going to be detected by imaging, okay? So high resolution cameras are going to capture the fluorescence emitted by each cluster during every cycle. The color of the fluorescence corresponds to the nucleotide that was incorporated. Like I said, maybe red for blue, sorry, red for A, blue for C, etc. Okay, so this is how it works. Step is the data analysis. These captured images are processed to identify the fluorescent signals from each cluster. So we'll analyze from each cluster what was the color of the fluorescent signals that we got. It's going to translate the signals into a sequence of bases. Okay, these raw sequences are aligned to a reference genome. Okay, so now that we got the sequence of our genome, we are going to compare it to a reference genome, okay? And we are going to see if there are any changes, any variations. So if any variations are detected in a any specific part of the fragment, we can come to a conclusion that this was the cause of the disease, okay? So this is how Illumina sequencing works. Was it clear till here? Anyone has any doubt in any of the steps? Any doubt in any of the steps, anyone?
Ma'am, could you please repeat the step 3 sequencing by synthesis?
Sequencing by synthesis. Okay. Okay, so in sequencing by synthesis, we are adding a primer which is going to bind to the adapter region of each fragment. So while we were adding the DNA fragments, we ligated both the ends with specific adapters, right? So we are going to add primers which are like short DNA fragments. They are going and attaching to this adapter. Okay, this is the primer. It has attached to an adapter, and we are adding four fluorescently labeled nucleotides, that is ATGC, along with reversible terminators plus DNA polymerase. Okay, fluorescently we are labeling the nucleotides to detect their emission. Okay, so to like detect which nucleotide has binded by the way they're emitting light, right? So that is one. Reversible terminators, they are added to ensure that DNA polymerase is uh, incorporating only one single nucleotide at a time. Okay, so these are all fluorescent nucleotides, right? So see, we've just so DNA polymerase needs nucleotides to replicate DNA, right? So we are uh, adding fluorescent nucleotides because we need to detect the sequence of the unknown sample, right? So when we add reversible terminators, it is ensuring that only, excuse me, just give me a minute. I'm just feeling a little sick. So, yeah. So we are adding label nucleotides with DNA polymerase. So DNA polymerase is replicating this DNA fragment by um, the incorporation of fluorescent nucleotides. Okay, so when we are adding only one nucleotide, when the DNA polymerase is adding only one nucleotide at a time, it makes it easy to detect, right? Rather than just adding a lot of nucleotides together, a single nucleotide is being incorporated so that we can detect the signals easily and analyze them easily. Okay, so the DNA polymerase is going to incorporate, see, only one nucleotide is incorporated at the time. Okay, so here T got incorporated and here A is going to get incorporated. So every time a nucleotide is getting incorporated here, a fluorescent signal is being emitted, as you can see, blue for A and green for a G and yellow for C. It's being emitted, right? So these specific colors, okay, are being detected by imaging and data analysis, and this cycle is going to be repeated for a number of times. Okay, so through this we are determining the sequence of our unknown fragment. Okay, I hope that was clear.
Yes, ma'am. Thank you.
Yeah, I'm so sorry for the frequent pauses. I'm feeling a little sick, so yeah. Anyone else? Any other doubts? Any other doubts in any step?
No, ma'am.
Okay, great. So let me just. Yeah, so this is a diagram which I just added for your reference. So these are the fragments. We add adapters to their ends. We are attaching them to the flow cell. They are binding to their complementary primers. They're going to be extended by PCR. Next, they're going to get dissociated into two single strands. Clusters are going to be formed. And then by the addition of fluorescent nucleotides, we are detecting the sequence. Okay. And we are scanning the signals emitted. Okay. So this is the difference between Sanger sequencing and Illumina NGS. Okay. So Sanger sequencing was the is a very old method compared to Illumina NGS sequencing. So Sanger sequencing is also, considered a bit dangerous because it involves use of some chemicals as well. Okay, so when you compare it to Illumina NGS, Illumina is obviously a more high throughput method, a more easy method, and at a time we are able to do this for millions of fragments. Okay, and the scale of NGS sequencing is much higher than Sanger sequencing. So this is just a difference between the Sanger sequencing procedure and Illumina sequencing. Okay. So the advantages of Illumina sequencing are high accuracy. So reversible terminator chemistry, like we saw, the reversible terminator, it allows addition of only one fluorescent nucleotide at a time. So that minimizes your errors, massive parallelization. So millions of clusters are being sequenced simultaneously. Scalability, which is suitable for small targeted experiments, to large scale whole genome sequencing, and it has a wide application, like it can be used for DNA sequencing, RNA sequencing, epigenetics, and more. Okay. Some of the limitations are, yes, because it is only for short red lens. Okay, so you can detect only like a specific number of base pairs. Okay, like for example, only till 50 to 300 base pairs, which may complicate the assembly of repetitive regions. So the equipment and reagents can be expensive, though the cost per base is low for large projects. So complexity. Um, to detect those fluorescent signals being emitted, we require a lot of computational resources to analyze the huge amount of data that is being put out by NGS sequencing. Okay, so this is about NGS and Illumina sequencing. I hope it was clear to everyone. When was my voice breaking? Okay, I'm so sorry. I think internet was not stable or my voice isn't stable. I just had a little cough, so yeah. Sorry for that. I hope it was clear how Illumina sequencing is done. I hope everyone understood. Any doubts? Anyone? So I just put out that YouTube video so that you can visualize it, because without visualizing, it'll be difficult to like understand the procedure. So anyone, any doubts? Okay, so okay. Okay, that's great. Okay, then this is all for today's class. Um, Tomorrow we'll look at some more sequencing technologies, and after that we'll have a practical session on how to analyze the data obtained from NGS sequencing. Okay, so this is all for today's class. Thank you for joining, everyone. Any doubts, you can ping me on WhatsApp. Okay.
Thank you, ma'am.
Yeah, thank you. Mm.